Viruses. You know, they are some of the most fascinating and kind of terrifying forces on our planet, right? I mean, these tiny little things have shaped, well, everything. Life, history, even our own DNA. So today, we're going to pull back the curtain and see exactly how they pull off this ultimate hijack.
And here's the thing that's always blown my mind. They're not even really alive. I mean, they're in this weird gray zone between living and not. So how in the world does something that's not technically living become the planet's most successful hijacker? That's the big question. Let's dive in and take a look at their playbook. Okay, so what are we really talking about here? At its core, a virus is just ridiculously simple. Think of it like this. It's a tiny little package. Inside, you've got some genetic code, DNA or RNA, basically its blueprint, and that's wrapped up in a protein shell. That's pretty much it. Now, scientists have this fancy term for it, obligate intracellular parasite. All that really means is that it has to get inside a living cell to do anything. It's a total parasite. Without a host, without you, it's just dead in the water. No engine, no way to make energy, nothing.
All right, let's get one thing straight right off the bat, because a lot of people mix this up. Viruses are not bacteria, not even close. You see, a bacterium is a whole, living, single-celled organism. It's got all the machinery to live its life, find food, and make copies of itself. A virus, though, it's like a stripped-down, bare-bones invasion machine. It has one job and one job only. Get in, take over. So, if you want to understand the hijacker, you got to understand its toolkit, right? Let's take a look under the hood at the basic anatomy of one of these invaders. Okay, so every virus has a few key parts. At the very center, you've got the genetic material. This is the instruction manual, the evil plan, written in either DNA or RNA. Then, wrapped around that, you have the capsid. This is its protective protein armor. And then, some viruses get extra sneaky. They actually steal a piece of the host cell's membrane on their way out and wear it like a disguise. It's called an envelope, basically a stolen invisibility cloak. And that stolen cloak, that envelope, it actually leads to two totally different strategies. The enveloped viruses, like influenza or HIV, they're stealthy, but they're also kind of fragile. Their fatty outer layer gets totally wrecked by soap and detergents. That's literally why washing your hands works so well. But then you have the non-enveloped viruses. These are the tough guys. They just have that protein armor, which makes them super resilient. They can hang out on a doorknob for a long, long time.
Okay, so we've seen the toolkit. A pretty simple package, right? So how does this minimalist little invader pull off what is, without a doubt, the most sophisticated heist in all of biology?
It all goes down in 6 main stages, like a perfectly planned heist. First up, attachment. The virus has to find the right target cell and latch on, kind of like a key fitting into a very specific lock. Step 2, penetration and uncoding. The virus gets inside and sheds its disguise, releasing its genetic blueprint into the cell. And now for the main event, replication. This is the hijack. The viral code takes over the cell's entire factory, forcing it to stop what it's doing and start cranking out viral parts. Once you've got all the parts, you need assembly. The new viral bits and pieces are put together, creating thousands of new virus clones. And finally, the getaway, release. The newly made viruses burst out of the cell, often destroying it in the process, and boom, they're off to hijack the next cell. The whole cycle starts all over again.
Now for that final getaway, viruses basically have 2 main playbooks. First, you've got the lytic cycle. This is the smash and grab approach. Replicate like crazy, burst the cell open, and get out fast. Maximum chaos, maximum speed. But then there's the lysogenic cycle. This is way more subtle. Think sleeper agent. The virus sneaks its own DNA into the host's DNA and just hides. It lays low, getting copied every single time the cell divides until some trigger wakes it up. Then it goes lytic and blows the whole place up.
And when you look at RNA viruses, the playbook gets even cooler. So, some of them have what we call positive sense RNA. You can think of this as a recipe that's ready to go. The cell's machinery can read it and start cooking immediately. Then you've got negative sense RNA. This is more like an encrypted recipe. The virus has to bring its own special decoder key to unlock it first. But the craziest of all? Retroviruses, like HIV. They don't just give the cell a recipe. They use a special tool to permanently rewrite the cell's master cookbook. It's just wild.
Okay, so that's the theory. But let's make this real. It's time to meet some of the most wanted criminals in the viral world. Let's look at their specific MOs.
First on our list, influenza. The flu. Its whole game is being a master of disguise. It never wants you to recognize it.
See, influenza's genetic code is a little sloppy.
As it makes copies, it's constantly making tiny little mistakes, or typos. We call this antigenic drift. And those little changes are enough that our immune system, which remembers last year's flu, doesn't quite recognize this year's version. That's why we need a new flu shot every season. But then, every once in a while, something much bigger happens. It's called antigenic shift. This isn't just a typo, it's a complete rewrite of the book, usually when two different flu viruses mix their genes together. And when that happens, you get a totally new virus that nobody's immune system has seen before. That's what causes pandemics.
All right, next up is HIV, the ultimate covert operative. This virus is playing a whole different game. It's not just trying to make copies. Its mission is to take down the very system designed to stop it, our immune system.
So how does it do it? Well, its signature move is an enzyme called reverse transcriptase. Remember that rewriting the cookbook trick? This is it. HIV converts its RNA into DNA and stitches it right into our own human genome. It becomes a permanent part of our cells. And its target is so precise. It goes after our CD4 T cells. Think of these as the generals of our immune system. HIV takes out the command structure, and without the generals, the whole army just falls apart.
And that brings us to a virus we all know far too well, SARS-CoV-2, the virus behind COVID-19. Its secret to success was simple. It was an absolute master at picking locks.
So the outside of the virus is covered in these things called spike proteins, and you can think of each one as a key. Well, it turned out that this key was a perfect fit for a lock found on a lot of our cells, a receptor called ACE2. Because this key fit this very common lock so perfectly, the virus could get into our cells with incredible ease, and that, as we all saw, led to a global pandemic.
But this isn't a one-sided story. The hijacked is only half of it. For every move these viruses make, we, as humans, have developed a countermove. So now, let's talk about how we fight back.
Our number one weapon, of course, is vaccination, and the science here is just amazing. We have a whole arsenal of strategies. You've got the classic approach, show the immune system a killed or inactivated version of the virus so it knows what the bad guy looks like. Then there's the newer tech, viral vector vaccines, which basically use a harmless virus as a delivery truck to drop off a single piece of the target virus. And of course, mRNA vaccines. These are incredible. They just send our cells the genetic instructions, the recipe, to build one tiny part of the virus themselves. It's like giving our immune system a training manual without ever letting the actual enemy in the door.
And to fight them, we have to find them first. We've got two main ways of doing that. The PCR test is like a crime scene investigator looking for the culprit's DNA. It's designed to find the virus's genetic code directly. An ELISA test, on the other hand, is like finding wanted posters that your own body put up. It doesn't look for the virus itself, but for the antibodies your immune system created to fight it. It tells you you've had an encounter, even if the fight is already over.
But here's where the story takes a really fascinating turn. What if we could use a virus as an ally? Get this. There are viruses out there called bacteriophages, and their whole deal is they only infect bacteria. So now, scientists are looking at these as a weapon against one of our biggest threats, antibiotic-resistant superbugs. You can't kill the superbug with drugs? Fine, send in a virus that's programmed to kill it for you. It's a perfect, the enemy of my enemy is my friend situation.
And that really brings us to the big takeaway here. For sure, viruses are the ultimate hijackers. They are this relentless, powerful force of nature. But as we learn more about them, from using them to fight superbugs, to even using them to deliver gene therapies, we're realizing they're more than just enemies. We are just starting to figure out how to harness their incredible microscopic power. So the question for the future really isn't just about how we survive them anymore. It's about how we might, one day, turn the ultimate hijacker into the ultimate ally.